Hello, everybody. My name is Marcia Higas, and it's a real pleasure for me to introduce today's speaker, Judy Campisi. Thank you all for joining. So Judy Campisi is today's speaker. She performed her doctoral studies at the State University of New York in the field of biophysics, and she subsequently performed postdoctoral studies here in Boston at Dana Farber Cancer Institute with Art Party, where she worked on cell cycle and gained her first exposure to cancer biology. She is a real pioneer in the field of senescence, and she first started working in this area since she was an assistant and associate professor at Boston University Medical School. In 1990, she moved her lab to the Lawrence Berkeley National Laboratory, and today she remains affiliated, and she is also a professor at the Buck Institute for Research on Aging. Judy is a true pioneer in cancer biology. She performed a number of landmark studies in the field of senescence, connecting senescence to aging, generating some of the first senescence biomarkers, and making fundamental connections between senescence and cancer. She discovered that senescent cells are not inert and generate a fascinating secretory phenotype known as SOS-P, which a coin which she coined. And most recently, she has discovered that senescent cells have a really fascinating metabolic phenotype. She's received a number of awards that are too numerous for me to read. When I asked her about what she was most proud of, instead of naming an award, she mentioned she was most proud of her lab and her role as a mentor, and I can speak personally to that. She's a wonderful mentor to not only her own lab trainees, but also other faculty and trainees in the field. She's an extremely generous scientist and person, and terrific role model for all of us. So I'll mention some of her awards for her. She's awarded the Longevity Prize from Ipsen. One university awarded her a Lifetime Achievement Award, and recently she was elected into the National Academy of Sciences. So I really look forward to your talk, Judy. Thank you so much. Before we transition to your talk, I just want to bring your attention to the website, and please go to this Dana Farber Harvard Cancer Center website so that you can view future talks as well as past recorded talks. And just as a reminder to the audience, please post your questions on the Q&A slot, and then at the end of Judy's talk, I can help moderate the questions. Thank you.
Thank you, Marcia. It's very, very nice of you to invite me to participate in this. This is very exciting. Whoops. Oh, okay, I can see. Let's try this. There we go. So this is just a disclosure. I'm not going to talk about much about the company, but I will start by talking about aging. And so what all of you know is nobody dies of good health, right? We all die of some disease. And this is a partial list of the many diseases of aging. Cancer is in a different font for a reason, which I'll explain in a moment. But what is striking about these diseases, very different tissues, very different manifestations, and yet all of them are extremely rare in young people. They all rise with approximately exponential kinetics, starting at about the midpoint of our lifespan, so around 50 to 60 years of age for humans. And those of us who work on aging, we're convinced this is not a coincidence. We're convinced that there are some basic aging processes that cut across all these diseases and all these tissues, and they drive what we recognize as aging. So as Marcia alluded to, we've been studying this cellular process called senescence, which I'll explain in a moment. And we think now that it's a pretty good candidate for being a basic aging process. I don't want to leave you with the impression that we think this explains all of aging. It almost certainly doesn't, and I'll actually show you some data at the end that says it doesn't. But we do think it's a major driver. And of course, that means that it's a major target for interventions. So many years ago, when I first realized that this process of senescence, which was first discovered in the 1960s by Len Hayflick, but it was really pioneers in the field like George Martin who really drove home the idea that this had something to do with aging. And then the question was, how do you go about identifying something that drives multiple diseases associated with aging? And so one way to think about the problem is, back to this slide, most of these diseases are degenerative. That is, the cells within the tissue and the tissue simply doesn't function as well. And at least from my perspective, I would be hard pressed to call cancer a degenerative disease. I think it is a gain of function disease. Most of these other diseases are loss of function diseases. Cancer can really be considered a gain of function disease. And so then that simplifies the question. And the question then is there a common biology that links cancer to the degenerative diseases that we associate with aging? And obviously, the way to go about answering that question was to delve into what we know about cancer. The cancer field has always been a decade or more ahead of the aging research field. And so when I was a postdoc with Art and as I started my own lab, quite a lot was known about what causes cancer. So what I'm going to do now is a gross oversimplification, but it sort of recapitulates what we were thinking about. So I think there's no doubt that mutations are at the heart of cancer. These are somatic mutations mostly. And we also know that that probably isn't enough. When I was recruited to Berkeley, I was recruited by Mina Bissell, who was a pioneer in the idea of the tissue microenvironment being a driver of malignancy. And this is actually from one of her publications in which she looked at normal breast cancer cells. So the red are the nuclei, the green is E-cadherin. They self-organize into these beautiful alveoli, and of course in A prime is breast cancer, and it's a mess. But she was able to trick these cancer cells into thinking they were in a different tissue environment. She did this with an anti-integrin antibody. It doesn't work for all cancers, of course, but she was able to pretty much revert the phenotype of these messy cancer cells to something that was more normal. So this, I think, cemented to the field that you need at least two things to make a cancer. You need the mutations, but you also need this tissue environment that encourages the growth of cancer. So here's the problem. I know, many of you know, that potentially oncogenic mutations start very early in life. I think it was Doug Brash who first showed p53 mutations in the skin of very young people. So we all are harboring potentially oncogenic mutations, even when we're young. And then those of you who are over 30, you know that tissue structure degrades with age. And we know then that the tissue microenvironment is also changing long before manifestations of disease. So the question then is, why does it take so long for us to actually see the clinical signs of cancer? And we think that the answer has to do with evolution, that organisms with renewable tissues were forced to deal with the cancer problem from the beginning, and they had to evolve mechanisms to prevent the development of cancer at young ages. And so we collectively call these tumor suppressive mechanisms. So many of you know that tumor suppressor mechanisms have been studied again. When I was in Art's lab, Ruth Sager was a pioneer in the field of suppression. She was actually the first to propose that senescence was a tumor suppressor mechanism. And now I'm going to argue that these tumor suppressor mechanisms actually came at a cost. And that cost is what we recognize as aging. So just to remind you that many years ago, Kinsler and Vogelstein, two other pioneers in the field of cancer, proposed that we could think about tumor suppression as coming in two main flavors, and they call them caretakers and gatekeepers. So caretakers are pretty straightforward. They take care of the genome. They help prevent mutations. They help repair DNA damage. They help prevent the things that cause DNA damage. And they can even be considered longevity assurance genes. They help assure our longevity by taking care of our genome. The gatekeepers are more complicated. They assess the state of a cell, and at least with regard to preventing cancer, they basically give a cell two choices. Either die, and it's clear why that prevents cancer. A dead cell cannot form a tumor, or undergo senescence. And I'll tell you in a moment that this involves an essentially irreversible arrest of cell proliferation. Again, a cell that can't divide can't go on to form a tumor. The problem is, over long periods of time, tissues that experience apoptosis will eventually undergo atrophy and degeneration, especially if it's the stem cell pools that are being eliminated. And the same thing is true of senescence. As tissues accumulate cells that cannot divide, then the tissue fails to undergo normal function and homeostasis. And I'll show you that the senescence response is actually more complicated and can actually be a major driver in a positive sense of what we recognize as an aging phenotype in tissues. So what is senescence? Well, the way we think about it is we think it's a tripartite phenotype. These phenotypes are usually linked, but sometimes they can be dissociated, but they're mostly linked. And they involve this, as I mentioned, this irreversible arrest of cell proliferation, this very complicated secretory phenotype, which I'll tell you about some more. And then the cells also acquire relative resistance to apoptosis. I want to point out we can kill them. I'll show you that we meaning the field, has learned of ways to get these cells to die. But in many ways, they're sort of like cancer cells, which also have relative resistance to apoptosis and yet can be pushed over the edge by certain types of therapies. So what causes cells to enter this complicated cell fate? Well, many of the things that do that are things that we recognize as drivers of both cancer and aging. So things that damage the genome or even the epigenome. Of course, many oncogenes will drive cells into this senescent state, but even things that cause changes in metabolism, mitochondrial dysfunction, for example, or organelle stress, like ER stress, can all cause cells to adopt this fate. The problem is that you would think then this is all bad, and what we should do is focus all of our attention on how to get rid of these senescent cells. And that is not as simple as we initially thought. And that's because there are good things that this phenotype does, and that makes sense. It was under evolutionary pressure. So I've already told you that the growth arrest is very important for tumor suppression. And we know this from mouse models as well as from humans that have mutations in the genes that impose this growth arrest. And those mice and those humans die an early death due to cancer. So you need this ability to stop cells in their tracks in order to suppress the ability of cells to undergo malignant transformation. But recently it's become clear that a wave of senescence is important for both certain structures in the embryo, and there the secretory phenotype plays an important role in optimizing the development of those structures. And parturition, there's a wave of senescence in the placenta prior to labor. And then finally, it's now clear that especially the secretory phenotype is important for many examples of tissue repair and tissue homeostasis, and especially in wound healing. And I'll show you a little bit of data about that. So really, the way we have to think about senescence and how it can drive the diseases of aging, including cancer, is that it's an evolutionary balancing act in which there are positive aspects and negative aspects. And it makes the idea of intervention a little bit more complicated than we initially thought. So let me just say a word about how we know whether a cell has entered a senescent state or not. There are many biomarkers now. There have been dozens of labs, and this is a partial list. But this is some of the biomarkers that have been used to identify senescent cells, both in culture, but also in vivo in mice and humans. And the good news is there are quite a few biomarkers. The bad news is absolutely none of them are specific. All of them have lives outside of senescence. And so it does make the field a little bit complicated when we talk about identifying senescent cells with disease and then attempting to control some of their bad effects. But using those biomarkers, many labs have asked when and where do senescent cells occur. And of course, they increase with age. To my knowledge, this is true for every vertebrate species that has been examined and every tissue that has been examined. With age, there's an accumulation of cells with these biomarkers. This is just one of them. It's an enzymatic activity, but there are many others that show this increase with age. The other place you see senescent cells frequently is at sites of those age-related pathologies when you compare diseased tissue to age-matched normal tissue. So here's an example from Claudio Torres's lab. This is obviously autopsy material. This is from a brain of a patient with Alzheimer's disease. And the senescence markers are primarily in the astrocytes. And I will remind you, it's the astrocytes that give rise to the common forms of brain cancer. But you find them in the lungs of, for example, patients with chronic obstructive pulmonary disorder, the major cause of lung failure in the elderly. Osteoarthritic joints, not rheumatoid arthritis, which is mostly autoimmune, but osteoarthritis, which is the age-related arthritis. You find chondrocytes that are senescent in those joints. And this just goes on and on for virtually all of those diseases that I showed you on that first slide. And you also find them at the sites of pre-neoplastic lesions. So in many ways, you can say that senescent cells are a smoking gun, right? They're present at the right time and the right place to be driving these multiple, various, disparate diseases of aging. The question, though, is what's the mechanism? How could they be driving such disparate diseases? And then, of course, do they actually do it? Because everything I've told you so far is this correlation. Let me point out that in no tissue is there ever a majority of cells that are senescent. I think at the top, there might be as many as 10% senescent cells in a diseased tissue or very old tissue. Most of the time, it's far less. It's 1%, 2%, or sometimes even less than 1%. So how could they be driving all of these diseases? And of course, we think the answer has to do with the senescence-associated secretory phenotype, or SASP. We tend to divide the SASP into two main categories: those that are p53 dependent and usually do not involve changes in transcription. And these include these damage-associated molecular patterns. We now know there are pathogen-associated molecular patterns that are also secreted by senescent cells. You would never pick these up doing transcriptomic analyses, but a large part of the secretory phenotype does involve changes in transcription. They are generally p53 independent. p53 tries to act as a brake to the secretory phenotype. And what both of these arms of the secretory phenotype have in common is that they can drive inflammation. And many years ago, Claudio Franceschi coined this term inflammaging to describe a feature of almost all aging tissues. And that is that there's evidence of low-level chronic inflammation, mostly as judged by infiltration of immune cells, primarily, although not exclusively, cells of the innate immune system. So everyone knows that inflammation, chronic inflammation, is not good. It eventually will destroy tissues because these innate immune cells are making all of these damaging molecules in an attempt to prevent a pathogen from inhabiting the tissue. Normal tissue functions are disrupted. Stem cells don't work well in an inflamed environment. And of course, inflammation greatly increases the risk of developing cancer. On the other hand, I will remind you, you'll never heal a wound if you don't have at least an initial short-term inflammatory response. So although many of these secreted factors are characteristic of an inflammatory response, I do want to point out that we tend to call this inflammation plus, because there are many things that senescent cells secrete that can't be really directly linked to inflammation. For example, a number of growth factors, and very recently we found a number of active lipids, bioactive lipids, that are produced by senescent cells. So we now think we understand how senescent cells can be driving so many different diseases, and it's due to these secreted factors. It's through their paracrine effects. And I'm just going to show you 3 examples, but there are many, many examples of this now in the literature. So the first, this is back to breast. These are mouse breast cells in a 3-dimensional culture. They self-organize into these beautiful alveoli. The red is beta-casein, so they make milk if we give them lactogenic hormones. And if we embed in these 3D cultures non-senescent breast fibroblasts, they maintain their function and their structure. And this pretty much falls apart if the cells we embed in these 3D cultures are senescent breast cells. This work was first done by Simona Paranello, and she was even able to identify the major factors responsible for this degradation of normal tissue structure and function. And it was mostly the proteases that the senescent cells are secreting. In this case, what we did was we took human neural stem cells, put them in culture. The green is BRDU incorporation, so they're proliferating. And then we took astrocytes, human astrocytes, and added conditioned medium from those astrocytes. And you can see if the astrocytes were not senescent, we still got lots of cell proliferation. If the astrocytes were senescent, there was something in there, we don't know what it is, suppressed the ability of those stem cells to proliferate. Ironically, when we isolated cancer stem cell from glioblastoma, this conditioned medium actually stimulated the growth of the cancer stem cells, even though it suppressed the growth of the normal stem cells. And then this work was first done by Anna Krutolova in the lab. She was working with a pre-malignant breast epithelial cell line. So they're pre-malignant, they don't form tumors. This is tumor volume over time. But if she co-injects them with senescent breast fibroblasts, these cells will convert to full-blown malignancy. This is in an immune-competent mouse, and this is what happens in an immune-incompetent mouse, but these are human breast cancer cells. And you can see if they're co-injected with senescent human fibroblasts, then the tumors are larger, and these tumors are much more vascularized than these tumors. So the idea is we're all accumulating senescent cells with age. They're secreting stuff and causing neighboring cells to fail to function, and this drives tissue degeneration. And of course, we know that we're also accumulating pre-malignant mutations with age. And so we would argue that senescent cells can also drive these pre-malignant cells into malignancy, and that can possibly explain some of the late life cancers. So you should be depressed. If you don't have senescence, you're going to die of cancer. If you do have senescence, you're still going to die, partly of cancer and partly of lots of other things. So, of course, what the field grappled with in the light of these data is, what do we do about it? And so I think the field, including our lab, came up with two main strategies, one of which has been abandoned, at least by us, although not entirely by the field. And that strategy is to stop the secretory phenotype. So what we've all done is we've drilled down and identified the pathways that drive these secretions. And we've been able to identify molecular pathways that drive the secretory phenotype. Good news is those pathways were mostly known. The proteins within those pathways have already been developed for as targets by big pharma and small pharma. And so there are drugs available to stop those pathways from operating. But here's the problem. These are at least 3 of the pathways that are known to drive the secretory phenotype. And none of these pathways are pathways that you want to suppress, certainly not chronically. Otherwise, you wouldn't be able to respond to DNA damage. You wouldn't be able to mount an acute inflammatory response when you're wounded, and you wouldn't be able to respond to growth factors and to nutrients. So these are 2 important pathways to have to suppress chronically for the purpose of preventing the development of disease. The other problem is you can add drugs to suppress these pathways. The cells stop secreting. You take away the drug, the cells start secreting again. So you need constant pressure from the drug. And of course, there's really no such thing as a perfectly safe drug. So we sort of abandoned this approach from an intervention point of view, although there's still lots more to be learned about mechanism, and went to the approach of attempting to kill senescent cells with the idea that it takes time for them to build up, so it would require only intermittent pharmacological approaches to get the cells to die. Now, the first attempts to do this was not done with drugs. It was done with transgenic mice. There are 2 such mice. One mouse was made by our colleagues at the Mayo Clinic, Jan van Dersen and Jim Kirkland, and then our lab. And the way we did it, and the way they did it too, was very similar. There were just different approaches with regard to what the killer genes are and what the visualization genes are. So what we did was we took a large bacterial artificial chromosome that contains the murine P16 locus, because P16 is a common but not exclusive marker of senescent cells. And so that's a disadvantage of all the mouse models, is they miss a fraction of cells that use, for example, P21 as a driver. But for many senescent cells, they're driven by P16. We then had the promoter drive a luciferase so we could image the cells, a fluorescent protein so we could sort them. But most important, this viral thymidine kinase that turns the prodrug anciclovir into a phosphorylated derivative that is a very potent DNA chain terminator. Senescent cells, of course, don't replicate nuclear DNA, but they do replicate mitochondrial DNA. And Remy Laberge was able to show that this phosphorylated derivative got into the mitochondria, fragmented the mitochondria, and the cells died by apoptosis. So between our mouse and the Mayo mouse, which is called the Incotac mouse, I would say now probably more than 4 dozen academic labs have our mice. And they then use those mice to study their favorite age-related disease. And I've listed, this is already out of date. I really can hardly keep up with this slide. It seems like every few weeks there's a new disease where it's been shown clearly that if you eliminate senescent cells, you can either ameliorate the intensity of the disease, sometimes you can prevent the development of the disease, and in a few cases, you can even reverse the disease. So this, I think, tells us that at least in a mouse, we can say senescent cells are indeed a driver of multiple and very different types of age-related diseases. So I'll just show you one example from our lab, because most of you are so interested in cancer, but that has to do with something we've been interested for a very long time, which is some of the adverse effects of chemotherapy. And now we're expanding this to some of the drugs that are being used to treat patients with HIV/AIDS. So many of you know that many anti-cancer drugs cause DNA damage, and they're effective. We also know that DNA damage is a major driver of senescence and the secretory phenotype. And we also know that there's a rising problem in clinic of people who have been treated with these DNA damaging agents. It cures their cancer. Don't get me wrong. These therapies work. They save the lives, especially of children. But there's a cost, and the cost are a number of adverse effects. And so we decided to try to test the idea that senescence was a driver of some of these adverse side effects. And so the first thing we did, and most of this work was done by Marco Di Maria, who now is a rising star in the aging field and cancer field in the Netherlands. So he treated our mice with these different chemotherapeutic agents. Notice some of them are genotoxic, some of them are just cytotoxic. And we always got an increase in the luciferase, indicating that there are senescent cells that are there. And so the first thing he did was he asked, what's the cost of having that burden of senescent cells? And here he used another breast cancer model. These are breast cancer cells in which a viral oncogene is driving the malignancy of these breast cells. And they express now a different luciferase. So now we're going to follow the tumor with firefly luciferase instead of senescent cells, which are marked by Ranella luciferase. So if you inject these cells into the mammary gland, and we do this usually into the inguinal mammary fat pad, they form local tumors, but they also metastasize to the lung and the liver. So the experiment is we give the animals breast cancer, we wait a few days for the tumors to grow, and then we give them the chemotherapy. In this case, it's doxorubicin. And then a few days later, and that's kind of important, it has to be within 2 to 4 days, we either give them ganciclovir to kill off the senescent cells or not. And we do that for only 5 days, and then we wait a few weeks. So here's a control animal, here's the primary tumor, here's a liver metastasis and a lung metastasis. Now, if we eliminate senescent cells from these animals after the chemotherapy, we have variable effects on the primary tumor, something we still don't quite understand. But we completely eliminate the ability of the cells to form metastases. And our hypothesis is that this is due to the fact that we've changed the systemic milieu, and those micro-seeded metastatic cells are no longer able to grow. And we have some evidence for that, although it's not totally proven. Another problem with especially the anthracycline drugs like doxorubicin is the cardiotoxicity. Of course, we can measure that in a mouse by doing echocardiography. This is a collaboration with Simon Meloff's lab at the Buck Institute. So you give the animals doxorubicin, and a few weeks later, they develop decline in ejection fraction and fractional shortening. If after the doxorubicin, we give them the ganciclovir, and this again has to be within a few days, we completely prevent that cardiotoxicity. We can't reverse it. If we wait 3 weeks and then give them ganciclovir, we can't reverse it. But what we can show is that you can prevent it. And of course, another problem is blood clots. And again, this is work that was done with Sulu and Pankaj Kapahi at the Buck Institute. So you can nick the tail of a mouse, and you can show that bleeding time declines, as does the hemoglobin content if you put the tail in some PBS. And doxorubicin makes the animal pro-clotting. And again, this can be prevented if you eliminate senescent cells. And we can show that this is really due to the fact that a number of factors that promote hemostasis are produced by senescent cells. So I think all of this says that we have pretty good evidence now that this secretory phenotype can be deleterious in a number of pathological situations. But again, coming back to the issue of why did this phenotype evolve? And of course, it really is because a lot of those factors are important for wound healing. And I'll just show you briefly some data. These data are published, and again, this is mostly Marco's work, Marco Di Maria's work. This is luminescence at the site of a wound. This is just a little punch biopsy on the dorsal flank. And you can see there's a peak of luminescence, and then it declines. And we can talk later about why it declines. But the other point I want to make is these are female mice which heal their wounds much faster than male mice. We don't understand this. We're actually trying to study this now. But we can prevent this peak by treating the animals with ganciclovir. So now we ask, what happens to wound healing if you prevent that peak? And the answer is wound healing is retarded. So here are the wild type mice. They heal their wounds in 5 to 6 days. Here are the animals in which we prevent that peak of senescence, and they're slower, and these wounds are also more fibrotic. Now, what Marco was able to do is he was able to sort the cells from the wounds. And what we know is that it's mostly the fibroblasts and the endothelial cells that become senescent at the site of those cutaneous wounds. So he could sort them, and then he could profile them and ask, what are they making? And we found, much to our surprise, that they're making this rather understudied isoform of platelet-derived growth factor. And he could now show these, so these are male mice, he can reverse that slow wound healing phenotype by simply providing those wounds with topical platelet-derived growth factor. So some of you should be puzzled because everyone knows senescent cells accumulate with age. I showed you that. They accumulate in the skin. I showed you some of that data. And wound healing does not get better with age. It gets slower. So how can we explain the data I just showed you with what we know happens with aging? And the answer had to do with a collaboration with Simon Melloff's lab, and this work was done by Michael Velarde, who has his own lab now in the Philippines. And what we did was we used a skin-specific excision of this antioxidant gene. This is a nuclear encoded gene, the SOD2 gene, but it's primarily mitochondria. And we were able to excise this gene in a conditional way by providing the animals with tamoxifen, which activated Cre. And when we do that, we get senescent cells in the skin. But here, those senescent cells are persistent. And now we can ask what happens to wound healing. And again, these are male mice. So here's the control. Here are the animals with persistent senescent cells. And now you can see wound healing is retarded. So the idea is that evolution probably selected for senescent cells to be transiently present for optimal tissue repair and possibly homeostasis. But with age, these cells persist for reasons that are incompletely understood, but probably has partly to do with immune decline. And now, because the cells are persistent, they can drive phenotypes. So this is good news. It suggests that there may be times and places where we want to eliminate senescent cells. Now, of course, we're not going to make transgenic people. So the question is, can we find drugs that do this? And the answer is yes. The field has been really quite active in developing what is now a relatively new class of drugs that are selectively called senolytic drugs. I should point out there's another class of drugs called xenomorphic drugs, which are designed to simply suppress the senescent phenotype. Now, many of these senolytic and xenomorphic drugs are synthetic drugs. Interestingly, a number of them are failed anti-cancer drugs, failed because of side effects. But many of them are natural products, and the natural products probably have more hope as a xenomorphic, but we're not sure about the senolytic effects of them. But nonetheless, there are drugs that will selectively kill senescent cells. I should also point out that the reason why a failed anti-cancer drug may work as a senolytic is because if you want to cure cancer, you have to kill 99.9 or maybe all of the cancer cells. Whereas if you want to reduce the burden of senescent cells, we know from the mouse models that you only need to eliminate maybe 70 to 80% of the senescent cells, even in a very old or very diseased animal. So you can get away with lower doses and less time of exposure to the drug. So, of course, the hope for aging is that we will be able to, I mean, there's still so much more basic science we need to do. So we need to understand what are the good factors and what are the bad factors. And then the idea would be to try to replace them in order to optimize tissue repair. But then in the case of diseases, we would hope to be very pointed and selective in how we eliminate senescent cells, targeting the cells better and also timing when we eliminate them better than we have so far. So this is, I think, this is the future. And I think it really is on the horizon. Let me just say a few things about the secretory phenotype, because of course, in order for any of these approaches to be effective, we need ways of, we need biomarkers that enable us to assess the burden of senescent cells in an organism, especially in a person. And so we think the secretory phenotype is the key. We recently published a very extensive characterization of the different SASP components that are induced by different inducers. This is an anti-HIV/AIDS drug. This is obviously an oncogene. This is a genotoxic insult. We've looked at soluble proteins and exosomes. We're now looking at the surfacome. And there is a core of proteins that's common among all of these inducers and goes across different cell types. And as I mentioned, we also now know there are a number of bioactive lipids. But what was especially encouraging was we compared our core with a recent publication from Luigi Ferrucci's lab in which he looked in the plasma of people. This was a horizontal study of biomarkers in the plasma of humans as they age. And there are about 40 proteins that overlap, and these are the top 4. And anyone who's interested can go to this website, and we're constantly updating this website as we uncover new proteins that are the secretory phenotype. And these are some of the bioactive lipids for those of you who are interested. Many of them encode prostaglandins and leukotrienes. So the other point I want to make about the secretory phenotype is that it's dynamic. This just shows you, this encodes a leukotriene. It's profibrotic. It has a very different time course of expression from, say, this prostaglandin, which is antifibrotic. And I should point out, when we look at patients with idiopathic pulmonary fibrosis, they show this pattern, but they fail to show this big induction of the antifibrotic prostaglandin. The other thing is that it depends on how cells become senescent. This is a big mystery still in the field, is with age, how do cells become senescent? How much is due to an oncogene, or due to DNA damage, or due to mitochondrial dysfunction? And all of these secretory phenotypes have their own characteristic depending upon how the cells become senescent. And then finally, I just want to point out this is single cell RNA-seq. So this is P21. This is the gene that encodes P21, one of the growth inhibitors that causes cells to stop dividing. So these are non-senescent cells. There's always a few senescent cells within the non-senescent population, but you can see that at senescent, there are many, many more P21 positive cells, but there are subpopulations that are somewhat devoid. Now, it turns out that many of these are P16 positive, but that's beside the point. Look at this graph. MIC-A is a ligand for natural killer cells, and we've shown that natural killer cells do target senescent cells that express this ligand. Very few non-senescent cells express this ligand. Many more senescent cells do, but again, there are subpopulations that don't express the ligand. So these are subject to immune clearance, and these would be deficient in immune clearance. Okay, so these drugs are on the horizon. We're going to learn more about them. They're going to extend our health span. What about, are we going to live longer? I just want to point out that experiment has been done in the mouse by Jan van Dersen. This is from his paper. What he was able to, so this is his mouse is called the Inkatak mouse. So his killer is different, but he can eliminate senescent cells from the midpoint of the lifespan until death. And he can show an impressive increase in median lifespan. These are two different strains of mice. No difference in maximum lifespan. So one possibility is, of course, something else is killing these animals. We need to know what it is, and then we'll all be able to live like Aubrey de Grey promises for 1,000 years. Well, the other possibility is that evolution is again teaching us a lesson. If you think about C. elegans, the world record is extending the lifespan of this organism tenfold. But as you get to more complex organisms, like even Drosophila, less than twofold. And then mice, well, even 30% is very rare. That has often been questioned. So it's possible that as complexity increases, evolution has to use more and more genes to set species-specific lifespan. And we may be very, very far from developing any drug that can do what evolution can do. So I'll just end by reminding you, this is the gold standard, right? This is Jeanne Calment. This is on her 121st birthday. She lived to be 122. Well, she was mostly blind, and she had osteoporosis and osteoarthritis. And I think this will be our model. This is Thurgood Marshall. He was the first Black Supreme Court Justice in the United States. Of course, it's a lifetime appointment, so someone had the nerve to ask him how long he plans to live. This was his response, and this may be something that maybe we can hope for. So let me stop by thanking fabulous people who are in the lab now, past lab members with whom we still collaborate, and many, many collaborators in many other institutions which made this possible. And I can stop now and take any questions.
Thank you so much. That was really exciting, and also really nice how you integrated so much history into the current work. So I'm going to start by reading a question from Marcelo. Can senescent cells influence the chemosensitivity of tumor cells?
Probably they can. As we learn more and more about the kinds of things that these cells produce, we know that they can produce proteins and proteases that can affect cell surface proteins on both normal cells and tumor cells. And that includes things that could transport drugs into the cells. So I think there's still a lot of work to be done, but my bet would be yes, they probably can. And it can probably go either way, depending upon what the phenotype, the secretory phenotype, is composed of and what dominates.
So Catherine Mittner-Dykhouse asks, you talked about P16. Have you examined the role of P15 in disease and senescence?
Yes, yes. So for a long time, we ignored P15, like much of the field did. We are now adding P15 to all of our examinations of old tissues and chemotherapeutic tissues. And I'll tell you an interesting, this is unpublished data, but the paper should be submitted soon. This was a collaboration with Luigi Ferrucci. So Luigi, being a clinician, is able to get an IRB approval a lot easier than we basic scientists can. And so he proposed that we do the following experiment, that he took a biopsy, a punch biopsy. These are young donors. A punch biopsy, small punch biopsy, and then a few days later take a second biopsy. The idea being to ask during wound healing in humans, do we see the same biomarkers that we see in mice? And to our surprise and his surprise, P16 was not very prominent, but P15 was. And so we now know that P15 is also another probably good biomarker and helps enforce that senescence arrest.
So the next question is by Sheila Stewart, and she asked Dr. Campisi, do you know if senolytics are equally efficient at killing age-related, oncogene-induced, chemotherapy-induced, SOSP-induced senescent cells?
Sheila.
First question, so I'll let you address that.
Yes. So unfortunately, the answer is not simple. It depends on the senolytic. We've worked with quite a few. We find that some of them, for example, are more effective at genotoxic stress-induced senescence. Others are more effective at oncogene or I should say RAS-induced senescence. And so it's probably going to vary with the senolytic. And this is where we have a big gap in our knowledge, and we need much more data.
So I'll let you address some of her additional questions in the text later, but I want to ask one of her follow-ups along these lines. Do you know how long, does it matter how long a cell has been senescent for?
Yes.
In order for the different senolytics to work?
So I don't know about for the different senolytics to work. We actually, some people, the field has now grown so much, so probably somebody knows the answer to that. We don't know the answer to that, but what we do know is that if we hold cells in culture for months, we see changes in gene expression that take literally 3, 4, 5, 6 weeks. And I believe John Sedevy has similar data with his interferon responses and the cytoplasmic DNA fragments that he sees coming off. So I would be very surprised if this was not also changing in vivo, and if it would not also affect how effective the senolytic could be.
So along those lines, there's another question about the timing and reversibility. So in terms of the irreversibility of senescence, do the cells re-enter cell cycle, or do they simply mediate cancer progression through SOSP?
I think that might depend on both the genotype and the inducer. So in the case of genotype, up until literally a couple of months ago, anytime we would induce senescence in a normal cell, we have never found any evidence of being able to get that cell to re-enter the cell cycle. Even if we come in afterwards and inactivate P53 or P16, the cells would rather die than go back into the cell cycle. However, and so let me also say, though, that a number of labs, though, have reported that in the case of a cancer cell that becomes senescent due to chemotherapy, for example, that they can, at least sometimes, can re-enter the cell cycle. Not all of them, but some of them. So it suggests that there may be mutations that prevent that growth arrest from being truly irreversible. And very recently, we found a drug that seems to induce senescence in normal human fibroblasts and endothelial cells. And when we take the drug away, it looks as though at least a subpopulation of those cells re-enter the cell cycle. So, you know, all bets are off in this field. It's changing so rapidly. There's no room for religion in this. We have to be totally agnostic.
So Philip Oberdorfer asks, you and others, your findings suggest that cancer cell responses to senescent cells are quite distinct from normal cell responses. Can you speculate why?
Well, they're mutant. I mean, I don't know of a single cancer cell that has a normal genome and it doesn't have derangements in responses to the environment as well as growth responses. So I think that's the simple explanation.
Okay, I want to ask one of my own questions. Can you comment on, have you examined how senolytics work, whether they work in animal models of metabolic syndromes and diseases?
By metabolic syndrome diseases, you mean things like type 2 diabetes?
Exactly.
Yeah. So how they work? Well, I can tell you what has been reported to work. And then based on what's known about the mechanisms of those drugs, you can speculate about mechanism. So the Kirkland group, or the Mayo group, I should say, because a large number of authors have shown that senolytics like dasatinib and quercetin can alleviate the senescent burden and some of the symptoms of type 2 diabetes, especially driven by high-fat diets. And we certainly see a preponderance, more senescent cells in age-related visceral fat, which does cause a decline, not strictly diabetic, but the animals do become more insulin resistant. And so we can show, for example, in that case that the ABT drugs, which activate the BCL2 family members, are also effective at eliminating some of those phenotypes as well as some of those senescent cells. But again, these are early days. So I think, and we need really more, broader range of senolytic drugs in order to be more specific.
Very interesting, thank you. I'm going to ask a question by Hugh Chuch Zan. He asks, is there any data that decreased senescence actually promotes stem cell aging, as this disrupts tissue balance and homeostasis?
Decreased senescence?
Yes. So you can comment on how does senescence
if you
stem cell aging?
Right. So if you deplete a tissue of stem cells, if you deplete a tissue of senescent cells, and some of those senescent cells are stem cells, does that have a bad effect on the tissue? Yes. So the short answer is it probably wouldn't make a big difference, because if the stem cell is senescent, it's not going to divide and give rise to regeneration. But I will tell you that with age, we also find, for example, in the skin, if we eliminate senescent cells in old skin, we do not improve wound healing. And the reason seems to be that they have already depleted a lot of the stem cells in the skin. So the stem cell problem is probably a separate problem, and it needs to be addressed by other means, I think.
Okay. Thank you very much. So because of the time, I'm not going to have time to read the more than dozen additional questions. I want to thank you guys all for posting your questions. Judy will have a chance later to type in the answers, so please go back and check the answers later. Thank you so much for a fascinating talk.
Thank you for the invitation, Marcia. Ciao to everyone.